Hi everybody. Today we will be discussing the tissue level and tissue organization. Basically, we're going through the organizational level. We started with chemical level, now we work through the cell, and right now we're at the tissue, which basically describes multiple cells with a specific function. So we have four different types of tissue: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue typically found as a covering, connective tissue as a has a supportive role. Muscle tissue role is movement, and nervous system function is control and coordination of your body activities. Starting with the first type of cells, your epithelial tissue, we have some main functions associated with them, including protection, absorption, and secretion, as well as ion transport, filtering, depending on what part of the body you can find them, and producing mucus to create a slippery surface for within the cavities or between the multiple layers of the body. Now, to connect the tissues and hold the cells within the tissue together, we use three different types of junctions. And we mentioned these earlier when we were discussing cellular level, so I will not be discussing this in more detail. But you do remember that we have three different types of junctions known as tight, desmosome, and gap junctions with very specific functions in the intercellular space. Now, what I'm going to focus on is on the epithelial tissue to start with. Epithelial tissue has two-part names. The first part of the name is depending on the number of layers that form the tissue, either a single layer of cells, which we refer to them as simple, or multiple layers of cells stacked on top of each other, which we use the word stratified to define it. The second part of the epithelial name comes from the shape of the cell. The shape of the cell can be categorized to either flattened cells, which we describe as squamous, cube shape, or under the light microscope, more of a square-shaped cells, which we refer to this as cuboidal, and cells that are column-shaped or elongated cells, which we use the word columnar to describe them. So basically, anytime I want to name an epithelial tissue, I will give it a first name, either simple or stratified, and one of the part of the last name, either squamous, cuboidal, or columnar. So in reality, I can have simple squamous, simple cuboidal, simple columnar. I can also have a stratified squamous, stratified cuboidal, and a stratified columnar. We also have a type of cells called pseudo-stratified columnar, which basically appears to be stratified, but it's just in appearance. So in reality, pseudo-stratified columnar would be one layer of columnar cells that appear to have multiple layers. Now, this is an idea of how the cell shape looks like. So these are your flattened cells in single layer. This is the squamous cells. And then if you have multiple layers of the cells, we refer to these as stratified. Cells under the microscope, under the light microscope, a squamous appears similar to that, flattened cells. Cuboidal cells appear more square-shaped, and columnar cells appear more rectangular shape. What you see on the bottom left-hand corner represents the pseudo-stratified cells that appear to have multiple layers, but in reality, as you notice, there is one layer of the cell, not multiple. Something else you should notice about this picture is the superficial aspect of the cell, the one that is basically either facing the surface of the body or lumen of any of the cavities, is referred to as the apical surface. And the layer all the way at the bottom, connecting typically to connective tissue, is referred to as the basal surface. You will get familiar more with this vocabulary as you start to draw your slides and get more familiar with histology. Here is when we discuss the concept of simple epithelia, so single layers of the cells. There are a couple of main functions associated with them. For squamous cells, the easiest way to describe them is diffusion. And for cuboidal or columnar cells, the two main functions are absorption and or secretion. Let's take a look at these tissues under the microscope. What you see on the top left-hand side corner represents your lung tissue, which also indicative of simple squamous cells. The gaps that you see right here or right here are basically the air sacs that are present within your lung, allowing for gas exchange. And the lining you see in purple represents multiple squamous cells that have come together to form your simple squamous epithelium. The darker spots that you see throughout, as you can see where I'm pointing out, these darker spots are identifying the nucleus of individual squamous cells. On the bottom left-hand corner, what you're observing is a simple cuboidal cells. Cells that appear to be a square shape with one nucleus, so single layer of the cells. So what you're viewing here as these circular patterns, these are basically found in your body. And this is a sample of your kidney for simple cuboidal cells. So this is basically four cells that have come together, for simple cuboidal cells that have come together to form these circular structures. Now let me note that even though you won't see the separative line for these cells, what you can notice is that these cells have a very distinct, relatively small amount of cytoplasm surrounding them. Therefore, we can identify them as simple cuboidal. That being said, as you can see in some of these pictures, you can see these lines that are forming the boundary of the cells as a plasma membrane, and it is visible on some, while on other samples it might not be as clear. Do keep in mind when I say something look cube shape or flat shape or elongated rectangular shapes, they are not perfect squares or rectangles. They're something that resembles as closely to those structures. The last picture you see on the right-hand side represents your simple columnar cells. What you notice is that there is a single layer, so one layer of cells with the lumen, the empty space in the middle. Notice that these cells are more rectangular in shape. They have more cytoplasm free, so you have one nucleus, not multiple stacked on top of each other, and cells are elongated. Therefore, this identifies as simple columnar cells. These cells can be found, or this type of tissue can be found in the stomach, in the intestines, as well as the iris of the eye to basically change the shape of your pupils, either dilate or constrict your pupil. This is a picture that is more similar to what you will observe under the microscope. The top layers that is appearing as a pinkish color with purple dots going through them as I'm running my cursor on it right here. These are the nucleus, and this area of the pink is representing the cytoplasm of the simple columnar cells. This is an example of your intestine. This is a picture of pseudostratified columnar. You don't really need to know this, so I will be skipping this slide. Now, if you have multiple layers of epithelial cells stacked on top of each other, you have the function, or you would get a tissue type called a stratified epithelium. Again, this will represent two or more layers of cells stacked on top of each other. A most important function associated with a stratified epithelium is protection. By just thinking about this, if you have multiple layers of cells stacked on top of each other, there is more levels or more protective functions associated with that. These cells at the basal layer have the ability to divide through the process of mitosis and regenerate the cells as the older cells are died off. A stratified epithelium can be found as a stratified squamous, a stratified cuboidal, and a stratified columnar. Now, in the case of keratinized, there is a protein that is found in certain parts of your body, including your skin and your hair, that is known as keratin. So cells that carry this protein, keratin, are referred to as keratinized cells and are very important in the functionality of your skin by waterproofing your skin. You also have cells that are considered to be non-keratinized, which means this protein is absent. And you can find examples of that within your esophagus, the vagina, which is where the cells are present. You have multiple layers of the cells, but the keratin, again, is absent. Most of these tissues do produce mucus for moistening and lubricating the surface of the organs. Here is an example of the stratified squamous cells. You do notice that many of these cells are flattened in shape, even though as you go more toward the basal layer, the cells look more cube-shaped, but majority of the cells look pretty flattened. This is an example of a stratified squamous, and this represents your skin. Now, the top picture you have here represents your stratified cuboidal cells. You can see there are multiple layers of cuboidal cells stacked on top of each other, describing the stratified characteristics. An example of this tissue is salivary gland. A stratified cuboidal is relatively uncommon in the body, so we don't actually look at it in the sense of histology when it comes to your lab later on. Another type of cells that we have in the sense of stratified is a stratified columnar, where you have these elongated cells stacked on top of each other. Extremely important for protection, but it also has a secretory role in the sense of production of mucus. An example for a stratified columnar is your urethra, or lining of the urethra. Now, within the stratified squamous cells, we have a specific type of cells known as transitional epithelium. As the word transitional implies, it means that it changes from one shape to another, in a sense, transitioning. It can be found in the bladder, in the urethra, which is a tube that goes from the kidney to bladder, and the umbilical cord. I will show you on the picture to make more sense of this. On the top, you see the picture of the stratified squamous, which we know as transitional epithelium. You have multiple layers of the cells, but these cells are relatively large and more cube-shaped. This is an example of the bladder. So when the bladder is empty, there is no stretching of the tissue, and the cells are relatively fat or cube-shaped. However, as the bladder starts to fill with urine, the size of the bladder starts to increase, and therefore the tissue starts to stretch. To accommodate that, the transitional epithelium cells also start to stretch, and you see the cells that are going from more of a cube shape to more of a flattened cell shape to represent the transition. Interestingly, as these cells, as the bladder is emptied, the cell revert back to their original relatively cube-shaped cell. For the purpose of our class, I will not be discussing a specialized epithelium, so you can glance through a specialized epithelium. You can skip the slide for glands, exocrine glands. You do need to know the examples, but you don't need to provide details of it, such as your sweat and salivary and oil glands. You don't need to know the unicellular exocrine gland. The only thing I do want you guys to know is the difference between exocrine versus endocrine. Anytime you hear the word exo implies outside. So exocrine glands are typically glands that are the glands that carry ducts that are open up to either inside the cavity or on the surface of the body, versus endocrine glands. These type of glands have no duct, so they're considered to be ductless, and they are associated with endocrine system that releases hormones directly into your blood, and therefore does not require any form of ducts to carry out the hormones from one location to another. You can skip the slide regarding the ducts. And what I'm going to do, I'm going to go through the slides for connective tissue, cover another topic, and then we'll come back and finish connective tissue for the next PowerPoint because it's relatively long. Muscle tissue, what I want to focus on is that there are 3 different types of muscles you have. These muscles are made up of, basically majority of them are made up of proteins. The 3 types are smooth, cardiac, and skeletal muscles. There are a couple characteristics you need to identify muscles with. One is a word used, striation. Striation is the light and dark patterns that are observed on the surface of the muscle if you're observing it under the light microscope. And a structure called intercalated discs, which are basically the type of junctions we identified as desmosomes that helps the cell basically connect to one another using linker proteins and enables the cell and the tissue to withstand the strenuous activity. Such structure was present in your cardiac cells to ensure that your heart can withstand the amount of work that it carries out every day. So let's start with a smooth muscle. A smooth muscle cells are a spindle shape, which basically implies that they are thicker at the center, and as you go on either end, they thin out. These cells carry a single nucleus because their size is relatively small, and one control center / nucleus is enough. There is no striation, the light and dark pattern, and there is no intercalated disc because a smooth muscle does not necessarily deal with a significant amount of pressure. Some of the locations that you can find these are, as I mentioned, iris of the eye, stomach, intestines, bladder, and uterus. Interestingly, the movement of the smooth muscles is identified as involuntary, which means that you have no control over such movement. Cardiac muscle, on the other hand, is considered to be striated. It does contain intercalated discs. It is typically carry one nucleus per cell, but at some times, depending on the length of the cell, they could have up to 2 nuclei. You cannot control the movement of this muscle, so it's identified as involuntary. And the only location you can find that is within the heart wall. Let me identify for you the difference between intercalated disc versus your striated pattern. So in this picture, what you see is the muscles are going vertically. What you see as these darker lines here, where the cursor is, here, or in the lower picture, the darker pink lines, are identified as intercalated discs, separating one cell from another. However, if you look closely, you also see a light and dark pattern going throughout the heart muscle. And these patterns you see in this region, going up and down, or in this picture going horizontally, are identified as your striated pattern or striations. Something else you need to know about cardiac muscle is the shape described as branched. As you can see here, these cells form multiple branches and can expand their size relatively larger than a smooth muscle cell. Last type of cells we have is skeletal muscle cells. Skeletal muscle cells are definitely long. They can get as long as 3 to 40 centimeters, which is about 20 plus inches, 18 inches. They do have, as I mentioned, a striation. Because of their length, they are always multinucleated. These muscles are under our control, so they're considered to be voluntary muscles. They do not carry intercalated discs, that is only and only limited to your cardiac muscles. An example of these type of muscles are any muscle that is connected to the bones via tendons. Examples, if you want to know muscle names, you could say biceps brachii, triceps brachii, your flexors, extensors, your pectoralis major, minor, etc. The striation pattern in the skeletal muscle is really clear. So if you look at the muscle going vertically on the top picture, you can see the light and dark patterns in purple color. that represents the striation patterns, and the darker purple structures between the cells are identified as the nucleus, and you can clearly see the multinucleated structures or multinucleated cells within the skeletal muscle tissue. So I'm going to stop here and we'll pick up the rest for connective and nervous tissue in the next PowerPoint.